Good morning everyone. I'm Rex Ryan E. Dahilan, and for today's presentation, I would like to present my exercise B, seed to seed, entitled Growth, Development, Reproduction, and Maturation of Mung Bean, Vigna radiata, with a variety of mat planted in pots. Mung bean or Vigna radiata L has gained prominence in sustainable agriculture and global food security efforts due to its nutritional value, versatility, and soil fertility enhancement through nitrogen fixation. Mung bean holds significant economic importance both locally and globally, contributing to agricultural economies with its versatile application across various sectors. The trend of cultivating mung bean seeds in pot has surged in popularity, driven by the convenience and adaptability of this leguminous crop. The compact nature of mung bean plant makes them well suited for container gardening, enabling individuals with limited outdoor spaces or urban dwellers to engage in home cultivation. The paper aims to explore the intricate aspect of mung bean cultivation in pots, specifically delving into its growth, development, reproduction, and maturation. The objective of this study is the study generally aims to evaluate the growth, development, reproduction, and maturation of mung bean, Vigna radiata planted in pot. The materials used in this study is mung bean seed, which is mat variety, medium for planting, which is pots, camera for documentation, measuring devices such as ruler, weighing balance, at lastly notebook where observation will be recorded. The procedure of this study is number one, each student is required to obtain the seed of his/her assigned crop. Number two, seeds will be planted in planting container and the experiment will be replicated 3 times. Each pot will act as a replication. 3 to 4 seeds will be planted in each container and will be thinned out 2 plants per pot for smaller crop, which is mung bean. A pinch of 14-14-14 or complete fertilizer will applied on each pot. Number three is another set of seeds will be planted in a petri dish lined with tissue paper. Students have to observe the changes that will happen until the plant emerge from the seed equivalent to plant that emerge from the soil. Observation will stop at this point for the seeds grown in petri dish, but observation will continue for the plants grown pots until maturity. Students has to write all their observations in notebook, indicating the date where they took their observations. The students need to document all their observation by taking pictures. To answer the question, what changes when a mung bean seedling grows? As mung bean seedling grows, various changes occur, including the emergence of leaves, elongation of the stem, and the development of fruits. These changes reflects the transition from germination to the establishment of a more complex plant structure. For the number two question, what special structures develop when a mung bean seedling grows and what is their function? So, special structures that develop during mung bean seedling growth includes cotyledon, which serve as the first leaves, and the primary roots that anchors the plant and aid in nutrients absorption. The cotyledon plays a crucial role in initial photosynthesis, providing energy for further growth. And for the next question, what similarities and differences are there among individual mung bean seedlings that grow under the same conditions? Individual mung bean seedling grows under the same condition exhibit similarities in their overall growth patterns, such as the emergence of cotyledon and the development of a central stem. However, differences may arise in terms of height, left counting, and branching, reflecting the inherent variability within the mung bean population even under uniform condition. Table 1 shows the days of emergence, number of seedling emerge, and percent germination of mung bean plants planted in pots. The table shows the germination and early seedling emergence study of mung bean. Vigna radiata L revealed noteworthy trends and considerations. In pot 1, all four seeds successfully germinated within 3 days of emergence, achieving a remarkable 100% germination rate. This outcome signifies the robustness and uniformity of germination in pot 1. In contrast, pot 2 and 3 exhibit slightly lower germination rates, with 3 out of 4 seeds in each pot germinating within the same 3 days period, resulting in an 80% germination rate. Despite the slight deviation, these rates still indicate a favorably and healthy germination process across all pots. After 1 week of emergence, a thinning process was performed, reducing the number of plants of 2 per pot. This practice aims to optimize the growth conditions for the remaining seedlings, ensuring adequate space and resources for robust development. The decision to maintain 2 plants per pot aligns with cultivation practices and promote healthier and more vigorous plant growth. To facilitate efficient data collection, individual tags were introduced for each plant in every pot after thinning. These tags serve as a convenient reference point for tracking and progress of individual plants throughout the growth cycle. The systematic approach of tagging allows for streamlined data collection, enabling researchers for monitoring and analyze key parameters such as plant height, leaf counting, and flowering patterns. So the table 2 shows the plant height of mung bean plants planted in pots. So one data plant per pot. So the table shows the dynamics of mung bean plants in 3 separate pots were observed over a 60-day period, revealing distinct ranges in plant height across different pots. On day 5, the initial height were 12 cm for pot 1, 11 cm for pot 2, and 10 cm for pot 3. As the study progressed, clear variation in growth patterns emerged. By day 60, pot 1 exhibit the highest final height, reaching 83 cm. Pot 2 shows a final height range from 79 cm and pot 3 range from 10 to 80 cm. So the difference in height ranges among pots suggests varying growth responses to the environment condition and practices. Pot 1 consistently outperformed the others, displaying a substantial range from the initial 12 cm to final 82 cm. Pot 2 and Pot 3, while showing comparable patterns, had narrow ranges from 11 cm to 79 cm and 10 cm to 80 cm. Respectively, the robust growth of Pot 1 indicates the effectiveness of the condition provided, potentially offering insight into optimal cultivation practices. So the table 3 shows the number of leaves in different pots. So the recorded data of number of leaves on the mung bean stems over a 60-day period reveals a consistent pattern of leaf growth across the 3 pots throughout the observation period. The number of leaves steadily increased, indicating a typical progression of a vegetative development. On day 5, each pot exhibit an initial count of 2 leaves, which is a common starting point for mung bean seedlings. By day 10, the leaf count had tripled to 5, suggesting a vigorous plant of leaf production during the early stage of growth. Between day 15 and 50, the number of leaves continued to rise, with minor fluctuation reaching a maximum of 22.6 leaves on day 60. This sustenance increase in leaf aligns with the plant maturation and canopy development, as mung bean plant tends to exhibit robust foliage during their growth cycle. It is important to note that the data presented here solely reflects the number of leaves that remained on the stems and does not count for any leaves that may have naturally fallen during the observation period. So table 4 shows the number of flowers per plant. So as you can see in the table, there is a variation of different flowers per plant as the day passes. So the table shows the number of floral openings in 3 distinct pots of mung bean Vigna radiata over a specific period, revealing intriguing insights into the plant's reproductive development. On day 31, pot 1 exhibit the initial floral opening, setting it apart from pot 2 and 3, which shows no signs of flowering at this point. By day 34, pot 2 and 3 caught up, each displaying one floral opening, while pot 1 continued to progress with 2 openings. As the observation period continued, the number of floral openings increased across all pots. By day 50, pot 1 led with 6 openings, followed by pot 2 and 3 with each 5 each. The trend continued until the final observation in day 55, where pot 1 demonstrates 7 floral openings, pot 2 had 5, and pot 3 showed 6. These findings highlight variation in the timing and progression of floral development among the pots. Pot 1 consistently exhibit an easier onset and a higher overall count of floral openings compared to pot 2 and 3. The observation differences may be influenced by a combination of environmental conditions and potentially nutrient availability. So table 5 shows the number of days to first floral opening. So the table shows observation of the days of first floral opening in 3 different pots of mung bean. Notably, pot 1 exhibit the earliest occurrence of floral openings, with the first blossom appearing on day 31. In contrast, pot 2 and 3 both shows a slightly delayed onset of flowering, with the first flower openings observed on day 34 in both cases. So in conclusion, the comprehensive exploration of mung bean research encompasses an economic significance, nutritional value, and adaptability in agriculture. The germination and early seedling emergence studies underscore the importance of favorable condition for optimal growth. Height variation observed over 60 days highlighted the potential influence of Environmental factors in mung bean development. Variability in the days of first flower opening among pots suggests the need for further investigation on the specific factors influencing reproductive milestone. The dynamic growth pattern in leaf count observation underscore the complex interplay of environmental factors. Collectively, these findings contribute valuable insights to refining cultivation practices and maximizing the potential of mung bean as a resilient and productive crop. So these are my documentation from the first appear of a different leaves up to the flowering and up to the harvest. So thank you for listening and watching, and Merry Christmas.